The work helps uncover a molecular explanation for recent observations that gut microbiome changes are associated with complex emotional behaviors. The research was conducted primarily in the laboratory of Sarkis Mazmanian, Luis B. and Nellie Su, professor of microbiology and affiliated faculty member with the Tianqiao and Chrissy Chen Institute for Neuroscience at Caltech. A paper describing the study appears on February 14 in the journal Nature. Decades of research have shown that the communities of bacteria that inhabit the intestines of animals, the microbiome, influence the immune system and metabolism. Studies in the last few years have linked the microbiome to brain function and mood. Further, studies in mice have shown that manipulating these communities can alter neurodevelopmental and neurodegenerative states, either ameliorating or exacerbating symptoms. It's been really difficult to show causation between something that's happening in the gut and the brain, rather than just associations between the disease states and the presence or absence of certain microbes, says Brittany Needham, first author of the new study and a postdoctoral scholar in Th. We were interested in trying to understand the molecular messages that are going between the gut and the brain, and how these signals may lead to changes in behavior. This study focused on a bacterial metabolite, a byproduct of microbes, called 4-ethylphenyl sulfate, or 4-EPS. Initially produced by microbes in the intestines, 4-EPS is then absorbed into the bloodstream and circulates throughout the body in both humans and mice. In 2013, the Mazmanian lab showed that this particular molecule was present in higher levels in mice with altered neurological development, specifically, a mouse model of autism and schizophrenia. Though other aspects of the altered microbiome differed from the healthy microbiome, 4-EPS levels were by far the most different. Additionally, in a screen of human blood samples from 231 individuals, 4-EPS levels were about 7 times higher in children on the autism spectrum than in neurotypical children. In this work, the team focused on the effects of 4-EPS on mouse models of anxiety. While anxiety disorders in humans are complex, animal models provide a way to study the precise changes in the brain and body that lead to anxious behaviors. Anxiety, in mice, is measured by their willingness to explore or hide in a new space as well as the time spent in a risky environment. The study compared 2 groups of laboratory mice. One group was colonized with a pair of bacteria that were genetically engineered to produce 4-EPS. The control group of mice were colonized with bacteria that were identical except lacked the ability to produce 4-EPS. Then, the mice were introduced to a new arena, and researchers measured each mouse's behavior. The mice with 4-EPS spent much less time exploring the area and more time hiding as compared to their non-4-EPS counterparts, indicating higher levels of anxiety. Brain scans of the 4-EPS mice also showed that some of the brain regions associated with fear and anxiety were more activated in addition to overall changes in brain activity and functional connectivity. Looking closer at brain cells within these altered regions, the team found that particular cells called oligodendrocytes were altered. These cells are important in part because they produce a protein called myelin, which acts as a protective coating around neurons and nerve fibers called axons, like insulation around an electrical wire. The team found that in the presence of 4-EPS, oligodendrocytes are less mature and consequently produce less myelin, leading to thinner insulation around axons. The basic framework for brain function includes integration of sensory and molecular cues from the periphery and even the environment. What we show here is similar in principle, but with the discovery that the neuroactive molecule is of microbial origin. Also, it will be critical to show that the human data have an effect in a well-powered and controlled clinical trial, which is now underway. In addition to Needham and Mazmanian, Caltech co-authors are former research technician Mark Adami, research technician Joseph Bakhtor, former postdoctoral scholar Wei-Li Wu, now of National Cheng Kung University in Taiwan, postdoctoral scholar Claire Rabbit, scientist Mark Ladinsky, lecturer in C. Additional co-authors are Masanori Funabashi of Stanford University and Daiichi Sankyo Road Novare Company, Zhou Wang, Yumei Guo, and Daniel Holschneider of USC, Gillian Haney and Daniel Geschwind of UCLA, Qian Zhu of UC San Diego and Arizona State University, Rob Knight of UC San Diego, and Michael Fiss. Funding was provided by the Center for Environmental Microbial Interactions, the National Science Foundation, the Human Frontier Science Program, the National Institutes of Health, the Ministry of Science and Technology in Taiwan, the Heritage Medical Research Institute, and Linda and Blaine Fetter. Explore the latest scientific research on sleep and dreams in this free online course from New Scientist. Sign up now. Brittany D. Needham, Masanori Funabashi, Mark D. Adami, Zhou Wang, Joseph C. Bakhtor, Gillian Haney, Wei-Li Wu, Claire Rabbit, Mark S. Ladinsky, Sun Zhong Wang, Yumei Guo, Qian Zhu. 2022, February 14. A microbial compound in the gut leads to anxious behaviors in mice. Science Daily. Retrieved February 14, 2022 from ift.tt/al9po California Institute of Technology. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.